at the University of Georgia, and did a couple postdocs, one at New York University and another at Stony Brook University, and came to USF in 2009 as a beginning assistant professor. And she's been an associate professor since 2016, and did a stint over in Germany on a special MOPCA GRI scholar at Tübingen University in Germany for a couple of years. And today she'll tell us about what she and her research group have been up to for the last several years.
Thanks, Steve. Really nice. I thought you weren't even going to introduce me, and you pulled all that off. Okay, so thanks for joining for this special Not Normal Tuesday seminar. I am going to try to cover a lot of ground, so I apologize if it's a bit speedy, especially if the thingy doesn't work. Okay. Yeah. Okay. So as Steve mentioned, I got my promotion to associate in 2016, and so it's been quite a few years, and we've done kind of a lot of stuff. So I'm going to try to squeeze as much of it in today as I can. The big context of the work that we do is really about biodiversity. So of course, most of this department is somehow concerned with biodiversity. Doing a presentation several years ago, I had this really fun task of going in and looking at all these really wonderful, diverse spots around the world to sort of highlight how biodiversity is pretty cool. So here's some examples for you. And of course, we have these common threats. When you Google threats to biodiversity, you'll get a list of 5 to 7 or 9, whatever your favorite list is. Climate change, invasions, pollution, destruction of habitat, overexploitation. Most of the people in our department are familiar with these threats to biodiversity and care about it. What you might not have thought about is that foundation plants in these ecosystems are really critically important. So even though they might not be threatened or endangered, like Spartina alterniflora, one of our favorites, understanding how those plants are able to respond to these diverse challenges is pretty important. And we argue that this requires understanding response to not just one environmental stimulus or challenge, but complex environments, right? It also requires understanding response of complex genomes, unfortunately, for some of us. A lot of these genomes can be really gigantic, have multiple ploidy, be the result of hybridization. We're able to apply genomics to some extent. I'm going to talk about how we've done that in our group. But first, I want to make sure that we understand that our work is still really grounded in classic approaches. So here we have Claus and Keck and Heisey. Hopefully everybody knows who they are. I'm looking at you, graduate students. Please know who these people are. Really did foundational work in California. Achillea is shown here. And what's really cool about this work is that they had sites all the way from sea level up to timberline, and they showed the diversity within the species. So we think often about cross-species diversity, but also within species, we get a huge range of phenotypes. And this is true for a lot of species. This is a different one, Potentilla, but what I love about this figure is what they've done here is they've gone to several different sites along that elevational gradient, collected rhizomes of the plants that they can then propagate and grow in common gardens. So down here we have Stanford, which is close to sea level, Mather, which is somewhere in between, and Timberline, which is right at the top of that gradient. And you can see that the different collections grown in these different gardens look really different. And you can see, for example, that this guy up here grows very well at Stanford, but as soon as you take him up to Timberline, he's not doing very well, right? So within species, we have diversity, and we have what we call adaptation to local conditions. They were able to show this because of clonal replicates across environmental conditions. And what's important here for our work is they show not only is trait mean, but also the plasticity. So the ability to respond here is the plasticity across environments. Those are both heritable, right? And therefore subject to selection and potentially contributing to adaptation. right? So from early in the 20th century, we knew already that genetics and environment drive phenotype. And that's really the foundation of our work. But at the same time, we know now, and even earlier in the 20th century, this is work of Jim Hamrick, who I worked with at University of Georgia when I was there in the Department of Botany. It was a real pleasure to be able to have that opportunity. His early work showed that across these different sites within this grass of Vena barbata, you can get associations of molecular markers, in this case, enzymes. So these are allozymes that are demonstrating some kind of selection. So different versions of the allozyme are associated with these mesic habitats, meaning more water, and different versions are associated with the xeric habitats, right? So this is basically the foundation of understanding how we use molecular markers to find associations with differences in environment. But it doesn't always work, right? So I really love I really love to point out Barbara McClintock's work, because early on, way before we had sequencing technologies, she was making these observations and telling us that a genome can react to conditions for which it's unprepared, but to which it responds in a totally unexpected manner. She's referring specifically to transposable elements. They can restructure the genome at various levels, from small changes involving a few nucleotides to gross modifications involving large segments of chromosomes. This was in her publication in 1984 after she got the Nobel Prize. This was way before genomics technology was widely applied, right? So really fantastic insight that we're still talking about today, trying to understand, right? So fast forward to my postdoc advisor, Massimo Pigliucci. He was also putting forward some of these ideas, that this idea that you can get from genotype to phenotype doesn't really work. Genes are not a blueprint to phenotype without the environmental context, right? So he's now in the Department of Philosophy in New York. He's always thought very philosophically, had a huge influence on my thinking. And working with him, we started to think about epigenetic modifications, okay? So you might see lots of different definitions of epigenetics. The one that we stick with is molecular level mechanisms. These mechanisms can alter gene expression and ultimately phenotype without a change in DNA sequence. There's tons of epigenetic variation that has no effect. So bear that in mind, just like sequence variation that we can't associate with a phenotype, also true for some epigenetic effects, right? So it's a big mess, but sometimes it has an effect. Sometimes epigenetic mechanisms can be triggered by the environment. Some of these changes can be inherited, right? So I know I'm hand waving a lot, which is really frustrating, but it's just the nature of the beast. The more we learn about it, the more we realize that it's hard to pin down. But part of the reason I really love these ideas is because of this concept that was published by Jonathan Wendell and Ryan Rapp in 2005 when I was starting to think about these ideas, that the genetic view here on top suggests that when you go through a bottleneck event, the number of genotypes reduces, right? So you lose the red genotype, and that translates directly into losing the red phenotype. So less genetic variation, less phenotypic variation, right? So these guys made this really nice argument that with epigenetic effects, you're still going to lose those red phenotypes that are associated with that genetic stuff, but you might get really unpredicted phenotypes. The ones that are in the A and the C groups here, for example. Still, this works nicely for summarizing the complexity of how epigenetics can be involved in things. So that was one very influential paper for me and my colleagues, Oliver Bosdorf and Massimo Pigliucci. We put a bunch of these ideas together and published this paper in 2008, where we made the argument that you have phenotypic variation, which affects ecology, and through natural selection, you get changes in genetic variation. And it's basically a cycle that we think about when we're thinking about adaptive differentiation. But now we're thinking that maybe there's other stuff that can contribute to phenotypic variation. Some of it can be heritable, so it can more quickly affect gene expression and phenotype, and therefore sort of accelerate this cycling in some cases. Turns out it's not so easy, but this was such a cool, fun idea in 2008. Still completely difficult to track down almost 20 years later. So just to bring home the idea of how we're trying to attack this problem, we have these molecular markers that I mentioned before with Hamrick's ideas, Hamrick's work. Tons of work has shown associations of differences in markers. So what I mean by markers is you're looking at a small portion of the DNA, but you can see that this version of that place in the DNA looks different, and it's associated with a habitat or a condition or a treatment, right? So what we can do to look at epigenetics is use different kinds of enzymes that are going to be sensitive to whether or not the methylation is sitting on some part of the DNA, usually associated with cytosines. Cytosines get methylated sometimes and other times not. So we can compare populations and see, for example, these two populations differ in their genetics. This population has no genetic diversity, right? Because all three of these individuals have exactly the same genetics, but they have different methylation marks, right? So that's the idea. You grow them also in common garden, because even though the genetics stay the same when you grow them in common garden, the epigenetics don't, right? So this is how we try to tease apart which epigenetic marks are heritable versus not. Good. So I'm going to tell you about 3 different big systems that we're working on applying these kinds of methodologies and approaches to try to understand response to different environmental conditions. The first is in salt marsh plants. This is the iconic Spartina alterniflora system. Those of you working in coastal systems inevitably would have encountered this kind of work. With climate change, we don't have much Spartina in Florida anymore, but along the East Coast, up and down the whole coastal plain, this is the plant that basically lives in the intertidal zone. It's a pretty spectacular plant, and the first time I saw it, I fell in love. We've done a lot of work in this system. I'm starting it for my dissertation, but I'm just going to talk about some of the more recent stuff that we've done. Here in Charleston Harbor, you can see the Spartina in the low marsh where it gets inundated but drains pretty regularly. Maybe not intuitively, it's taller right on the edge of the estuary. As you move in the estuary, into the estuary, like upland, the plant is more stunted. That's usually associated with accumulation of salt, anoxic conditions, etc. The big debate in this field, which is always interesting to think about, is whether or not these populations are actually genetically differentiated. For decades, many ecologists assumed it's just all the same grass. And what we did recently was to collect the short form and the tall form seeds from these different populations, and they're replicated across the landscape, right? So you can go to lots of population, collect these seeds. So here you can see how different they are in the field when you actually measure them. They're pretty different. We grew the seeds in common garden to show that, in fact, the tall form seedlings stay taller than the short form seedlings. And then we transplanted them into the field, sort of the gold standard of looking to see, are they actually differentiated when you put them in the environment that matters? And this might not look like a very exciting graphic, but it in fact is really cool, because here you can see in the short transplant zone, you get the short plants are doing better than the tall plants. Yeah, exactly. And then in the tall transplant zone, there's a crossing of this reaction norm, right? So this is indication that the plants do better in their home site, which is sort of our standard local adaptation. For those of you who think about fitness, how do you measure fitness? How could you possibly capture fitness? This is using what's called the Aster model. It's a package in R that you can use. It's also, you can do your own version of Aster, but it's combining survivorship and seed production into an integrated fitness. So this was a big finding for salt marsh ecologists because there's been support for both sides of the argument, but this was the first time we really had reciprocal transplants in the field. We also used molecular markers to show that this pattern of differentiation of tall from short form origins, we have collections from up and down the East Coast that show with genetic markers that the red guys, which are the short form origin, and the black guys are actually differentiated. So you have this pattern across several different sites on the East Coast. Then our fearless graduate student, Jeannie Munger, along with other postdocs and graduate student Sandy Vors, applied the molecular markers that allowed for her and them to look for methylation differences associated with these different habitat types. And in this case, I can't go into the details, but we used a matrix regression approach to show that we can predict phenotypic distance based on genetic, epigenetic, and habitat differences. Okay, so this is published in Frontiers, but basically summarized in this table that not only do the genetic differences associate with phenotype, but also the epigenetic differences associate with phenotype, and the actual physical environmental distance associates with phenotype. Right, so these are the locations of those populations in Charleston that Jeannie worked on with lots of help from others. Lauren Walling, who is here in the audience, is now comparing these populations to invasive plant populations in France and using the data that we have already collected here to see that the genetic variation is in fact reduced in the invasive range, but there's more epigenetic variation, and that is still a work in progress that should be coming along in the next few months. Another set of studies in this same system involved the Deepwater Horizon oil spill in 2010. As I mentioned, this is the main foundation plant of the coastal intertidal zone in lots of the US. Here you can see where the oil came ashore, and what you can actually see in this picture, if you look closely enough, is that these guys are all dead, but it's above ground that's dead, and you can see these little green guys coming back. And so the question was, are those green guys particularly tolerant, or what's happening to the population structure, or is there an impact? So this started with a microarray study. So this was early on, right? We went in 2010 to collect these plants. Microarrays was still a thing back then. Many of you probably don't remember what that is or ever knew, but microarrays were really cool slides that had probes for specific genes, and you could build a specific slide for whatever species you happen to have sequence data for. Luckily, we have these colleagues in France who are geniuses, really, working with this complex hexaploid plant. We're able to make a microarray for us. We use the microarray to show variation in gene expression between the oiled and no oiled populations. Over here is the design. We had almost 16,000 genes that we interrogated. And you can see here that oil exposure explained about 25% of the variation in gene expression. And what was really cool about this study is that there were also these excellent colleagues over in France who were working on this so-called xenome concept. So they had these 5 families of genes that are involved in the detoxification of the tissues in plants. And we were able to compare these 5 families of genes to published data in Arabidopsis that had been exposed to phenanthrene, which is one of the major toxins in oils. So Arabidopsis, for those of you who don't know, is the model plant. We have tons of information and resources. We know a lot about the genome of Arabidopsis, and it's the one that you would go to compare because it was the only plant that we had that kind of information for at the time. And what we discovered was that there's not very much overlap between which genes respond to this stress in Arabidopsis compared to Spartina, which is actually not that surprising considering Arabidopsis is this tiny dicot with 5 chromosomes. Spartina is a massive grass with 66 chromosomes, or 60 chromosomes. But what's interesting is that you can see not only do they not overlap, but they're basically different genes responding, right? So you can see here in the left column, those are the genes in Arabidopsis. The red is which are upregulated, which are downregulated is black, or yeah, not different is black. And so you can see the comparison of what's happening in Arabidopsis to the comparison of what's happening in Spartina here across all these different genes. And there's basically not much in common. And what's also really fascinating here is this massive amount of genes that are upregulated in Spartina, specifically in these detoxification pathways. So Spartina seems to be particularly good at dealing with this kind of toxin, maybe because it's got lots of copies of genes to bring in to the equation. So again, we were interested to think about if we could find epigenetic patterns here. You can see here on the left that the oiled populations, this is an ordination, PCA type of ordination. You can see the oiled populations separate out from the no oiled populations, both in genetic markers and epigenetic markers. Oil, again, here is explaining 23% of the variance in genetics, 17% of the variance in epigenetics. But here's the problem. So when we start using this type of technology, we can't easily map from the sequence differences or the methylation differences to the transcriptome. So the transcriptome gives you the actual genes that are differentially expressed. This is based on reduced representation across this gigantic genome. So it turns out that even though we have differences between oiled and not oiled plants, we can't figure out what those differences are associated with in the genome, right? So this is one of the biggest take-homes of the last 10 years of my life. We have a lot of limitations when it comes to trying to approach ecology with reduced representation sequencing, which is what we have to use because we can't sequence the full genome of all these plants. It's particularly challenging to link polymorphisms of either genetics or epigenetics to actual function. Okay? So we'll come back to that. I want to briefly tell you about some work in mangroves. We haven't been able to get very far with the system yet, but we have had a couple of really cool studies. And part of the reason that mangroves are so cool to work with when you're thinking about genomics is that the propagules germinate and mature on the maternal tree, right? So you can go out there and isolate your favorite trees, pluck off the propagules, and know that they're at least half SIBs, right? So it's a natural quantitative genetics design without so much work, which is helpful because everything else about working with these plants is challenging, right? So Kristen was a master's student working with us, and she collected plants from around Tampa Bay with that in mind, knowing who the mom was and having replicates of propagules from each mom. She had a gigantic experiment in the greenhouse, 1,200 plants from 6 populations, 10 maternal trees, and she had salinity and nitrogen bacterial combinations. This was in collaboration with Dr. Lewis. A really cool study. And what we found is that both population and maternal family being which tree they're from, explain variance in survival, as well as all of the traits that we measured. So depending on who's the mom, you're going to get variation in basically every trait, right? So you can see that here with succulents being how many grams of water is in a leaf. So that's sort of usually responding to salinity, but not in this case. Here you can see the interaction for root to shoot biomass allocation, which depends on what treatment of nitrogen you have, how the different populations are going to be responding, right? So this is a really nice way to try to dissect that there's variation in these important traits, which could potentially contribute to response to future environmental conditions. So I'm not going to go into the details of any of these studies. I apologize for that, but I just want to give you sort of the highlights. Then Jeannie, who was working in the lab at the time, was able to use reduced representation markers to ask again if the genetic and epigenetic variation is structured by population or the maternal family, and how much of the offspring variation is explained by the mom, okay? So here we found, which is known in the literature, is a really fascinating story about how mangroves have only recently sort of invaded the coastal habitat. There's not a ton of variation in most mangrove populations. Ours is very low, and it's structured by population and maternal tree. We also found that epigenetic variation is much greater. So this is hard to see because unfortunately the scales are not the same, but actually the range of variation in the epigenetic variation here is much higher than what you have in genetic variation. And again, this takes advantage of that quantitative genetics framework. So what we can see is the mom explains about 26% of the methylation patterns in CG contexts across the genome. So this is one of the first pieces of evidence that we have inheritance of methylation patterns that depends on who your mom is. Okay, so I just wanted to give you a little brief highlight about that work. We had students working on those projects for several years and it was very cool, but we haven't been able to continue it yet. So I'll have to come back to you with more on mangroves. Okay, so finally I want to talk about the invasive plant work that we've been doing in Japanese knotweed, which is the MOPCA work that Steve referred to at the beginning of the seminar. And the context here is a little bit different than the foundation plants like Spartina and mangroves, because in this case, we expect that there's not very much genetic variation in invasive species, right? You have a cool plant in Japan, for example, that Van Siebold, in this case, brings to the Netherlands because it's really cool, it's very exotic. He brings a few individuals. It turns out one genotype ends up being taken to London and then the New York populations. There's also another case where somebody sent a knotweed plant directly from Japan to New York. So some of this is really fun because you go into the historical documents, people writing letters to each other about how cool this plant is, like old horticultural appreciation. But what happens here that's really fascinating with lots of invasives, and Japanese knotweed is really iconic for this, is that it looks like a pretty normal plant when you're in the forests in China. But when you get to Europe and the United States, it becomes quite the monster. So eating people, basically. I mean, this is an undergraduate student who is doing field work for us when we started the MOPCA project. And that is a knotweed plant that we were digging up. So we were digging these up and sending them back to China, measuring stuff in the field, etc. So early work showed that the invasive populations all had the exact same genetic fingerprint, right? So here we have each lane is a different individual. All these markers across this gel show different fragments. And you look across the gel and you see they're all the same. So this was lots of different labs and different types of markers, isozymes, rapids, AFLPs, including the work that we did in New York, where we showed across different habitat types, it was the same genotype. So some of those habitat types are here. And of course, after working in the salt marsh in Georgia for my graduate work, I was amazed that this plant can live in the salt marsh. It also lives right in the middle of the beach on Long Island. These are on the northern shore of Long Island. It lives all over the place on roadsides and along these vast stretches of rivers, really fantastic populations in the Carolinas, for example. So is it the same genotype accommodating this huge diversity of habitats? The MOPCA project was funded by the German government. Basically, the academic exchange was the people who were managing the money. And what it allowed us to do was to set up a giant collaboration with people around Europe and people in China. We sent out 3 teams one in Europe, one in the US, and one in China over the summers of 2019 and 2020 to collect along these 2,000 kilometer gradients. And we collected traits and soil in the field, and then we replicated the rhizomes by cutting them up and sending them to China. So we had 250 individuals in 50 locations along each of those transects. And then we measured traits, we did genetic markers, we have soil and climate data. So I'm going to present some of that work, some of which has just come out in the last year. And here are like a handful of the people who worked on this stuff. This is the first time I've had 5 first authors on a paper, but the journal let us do it. So you know, if it happens to you. So these figures show you the results of growth, defense, performance, how they vary across the native and introduced ranges. So the red guys here are the Chinese populations. Then you have blue, North American, and Europe. You can see that China in general has lower standing biomass. So the way this is like a density distribution. So what's happening here is that the peak of the Chinese populations, most of the Chinese populations have a lower standing biomass than the European and North American. Same is true of leaf area. And then chlorophyll content's not quite as obvious. You can see that the European populations are a little bit lower than the US. There are other traits in the actual paper. This is just to give you an example of what we did. What was really fascinating is that we found a big difference in the amount of leaf area lost to herbivory here on the left. The Chinese populations have much more herbivory going on, and pathogens presence was really very strange because none of us are thinking about pathogens, but we found that the evidence of pathogens was much higher. The majority of the plants in China had some kind of evidence of pathogens, whereas we did not see that in Europe and North America. So with other traits that we measured, we were thinking about this combination that people often think about with the leaf economics spectrum. This is a big paper for people thinking about physiological ecology in plants that was published 20 years ago now. And in that paper, they made the case for 2,500 species across lots of different sites, and they came up with this sort of summary that plants often are either slow growing and conservative with their resources, or they're fast growing, they rapidly take up resources, and they're less efficient with it. Other people added to that this idea that leaf chemical defense and seed size can also be part of this equation. And we're talking not just across species, but also within species. So this is the context we were trying to think about for knotweed. Is it so different in the native range compared to what it's doing in the introduced range? And what we found is that it actually is different, which is really fascinating because I don't think about chemistry that much. But this is the great thing about working with a giant group of people. You have so many really cool ideas that you never thought about, but actually turn out to be quite interesting. So these figures here show you a bunch of the different traits that we were able to measure for the chemistry and the biomass. So we have flavonoids, lignin, alkaloid, tannins, toughness, chlorophyll, carbon to nitrogen ratios. And what these plots show you is as you come further out on these spider plots, you have higher levels of these kinds of traits versus, for example, lower levels of alkaloids, biomass, and SLA in China, right? So in China, we have one cluster, if you will, of plants that look like this with low biomass and low nutrition, but they're well defended, right? Because they're tough. They have tannins, they have lignins, flavonoids. We also have another type of plant in China that has lower defense. We call this intermediate because it's kind of like, what is it even doing? We don't know for sure. Wasn't as common as the blue guys, but here we have low biomass and lower defense. Whereas in Europe, what we found is different strategies. So these green and red strategies, high biomass, low carbon to nitrogen, high SLA and flavonoids, or intermediate biomass, low carbon to nitrogen, high leaf toughness and tannins. And then in the US, we have both of those strategies, but we also found another strategy, a fifth strategy with intermediate biomass, low carbon to nitrogen, high SLA. So we summarized that in the Global Ecology and Biogeography paper that came out a few months ago. It's a very cool looking figure, but it also shows you the differences between China, which is over here barely on the page. The types of strategies we found in China are pretty different from the strategies we find in Europe and North America. So kind of a surprising result for me. I didn't have any idea what to expect, but it turned out pretty well. So we also looked at the environmental conditions, which differ between the native and introduced ranges, also kind of surprising. So China is separating out on PC1 from Europe and North America, but even Europe and North America don't overlap that much, right? So that's really interesting. So all the ranges are different, but China is even more different. Remember, the PC1 explains more of the variation in the data set than PC2. In terms of soil, there's a lot of overlap. We couldn't really use it for detecting major differences between the three ranges, but there it is. And really surprisingly, we did not find good evidence that the environmental differences explain the variance in these traits. So again, we have biomass, specific leaf area, leaf chlorophyll. Most of the models that we ran, and this took months running all these different kinds of models, most of the models only explain 10 to 20% of the variance in traits. Most of the models that have environmental conditions, right? So that was still a big question mark. Coming back to the stuff that we really care about, though, how much of the differences in the traits that we detected is actually heritable? This is a really rare opportunity because we were able to send all of those rhizomes to China, right? So what's shown here are two gigantic common gardens. One is in Fudan, which is Shanghai. The other is in Xishuangbanna Tropical Botanic Gardens, which is in the Yunnan province. And we had tons of help running these experiments, and basically total 566 rhizomes. So rhizomes are individual collections from the field that we can't really identify if they're different genotypes, but we break it down by rhizomes. And one really exciting result from this, as a person who's been studying phenotypic plasticity for most of my career, is that we found differences in plasticity. But what you can see from this figure is that the Chinese plants here, the blue guys, are taller, have a bigger diameter, but have fewer ramets than the introduced populations, right? And we have Japanese plants here because we have colleagues who sent us some rhizomes from Japan. And this is just stunning to me because the Japanese and the introduced plants basically look the same. And we identified these Japanese plants as the likely source, so that's fair, that is not so surprising. But what isn't the same is the number of ramets, right? So my doodad is stuck. The number of ramets is this last panel over here. And it might seem a bit subtle. I think I have a problem, Houston. Hi. my advancer is not advancing. I don't know. I don't know where the mouse is. Try this. We're here. You're gonna break it. Oh, is it working? Oh, fine. Fine. I'm fine. You're fine. We're all fine. Right. So this is for our lab, this is a very exciting result, right? Because when we're talking about plasticity, we're talking about slope. And when we argue that plasticity is different among groups of individuals, we're basically arguing that the slope is different. Okay, so you can see this. It might take a little trust, but here we have Japan. The plasticity of the Japanese ramets is a flatter slope than the plasticity of the North American and European plants, which is really interesting because there is quite some discussion about whether or not plasticity would evolve in the introduced range and how that could be an important trait. Also really interesting because we don't think this plant should have any variation in the introduced range. So how does that work? Well, that's a question that we don't actually know, but it turns out that we do know that the plants from Japan that we have share the exact same haplotype as the plants from the introduced range, right? So we were able to run chloroplast DNA markers on all of these plants, thanks to a very motivated graduate student in China. And the introduced Raynaudtia japonica, this is the fancy genus species for knotweed, shares a haplotype with plants from the southern island in Japan down here. So you can see this big collection of blue circles here is the same haplotype as what we find in US and Europe. Exclusively in Europe, there's one different haplotype that we found in the US. And when you look at the phylogeny, this is what that looks like. It's actually quite interesting because here's all of Japan down here. There's some diversity in published chloroplast genomes of different Japanese knotweed. And then here's ours, our H45 that we collected in the population we think was the source, which is exactly the same haplotype as the North American and European populations, right? And then there's this unfortunate guy up here that we don't know anything about, but that was only one population. Okay, so we found the source of the introduced populations, we think. We're also looking at diversity within the introduced populations, and we found using RNA-seq SNPs, so we're running an RNA-seq analysis, but also looking at the SNPs within the RNA. You can see here, this is recapitulating that pattern of here's China over here, here's all of US, Europe, and Japan in terms of polymorphisms within the RNA. Right. However, we have not been able to find association with herbivory, which was the point of this RNA-seq. And we think it's largely because of this problem here. China versus Japan, there's 42% differentially expressed genes. But when you compare Europe to Japan, there's 3%. When we compare US to Japan, there's only 0.04% of the genes are differentially expressed. So these are sort of standard genes. We haven't gotten very far with this analysis yet. But this is one direction that we're trying to go with this work to get at which genes are differentially expressed in response to herbivory. So you'll hear more about that in the future. We've also been able to run the GBS SNPs in this global sample, which supports a Japanese origin. You can see here, Japan is this little tiny purple thing here, and then Europe and US has a bit more variation than Japan, probably because we sampled in a really small location. But we're not sure exactly. And then there's China over here. So these are the different SNPs that we find in China versus US and Japan. But this finding here is a bit alarming for those of us who thought we were working with one giant clone across this landscape, right? Because this still is showing us a lot of variation. And this sent us down a very long rabbit hole. I think it was years of our lives collected together. Trying to figure out how many polymorphisms do you expect to find within a given genotype, right? So you take the same sample of DNA and run it twice on the sequencer, you get polymorphisms. Like, this is the bad unwritten part of the story of sequencing, right? Like, we all thought this was going to solve everything. Like, we get to sequence stuff now, we can know the answer. But the truth is that it's still a game of statistics. So here you can see these are technical replicates. literally the same individual run on through the protocol on the sequencing. And this is how much variation you get. Like, that's not zero, right? So where is zero here? This is not zero. Like, look at all this. These are technical replicates. So, not to make you afraid of using this technology, but it makes me afraid. Like, I'm losing sleep over this problem. So what we've had to do is come up with a cutoff that says if there are this many polymorphisms, they're likely the same individual. And it's only these guys that get above that cutoff that are probably different. And what's unfortunate about this finding is that these red guys are the Chinese guys, populations, like comparing within populations. So within populations, they're also probably just the same individual, which is no different from what we find in the introduced range, despite our big storyline. So that's just an insight to a little bit of my pain and suffering in this pursuit of work. It's still very cool. This guy lives in really different places, somehow accommodating really different environmental challenges. But is it mutation? Is it accommodation of those mutations into the genome? What's actually happening over evolutionary time is still a really fascinating question for us that we're trying to chase down. So to this point, the main take-homes are that plants in introduced populations are larger larger in terms of biomass, with novel ecological strategies. We have pretty good evidence to that. But we were comparing those plants to China, so that's a big problem. We don't have data from the Japanese populations. But we did confirm the same haplotype with Kyushu, Japan. And introduced plants are still remarkably similar to those plants from Japan, except for this one finding of plasticity. And I have to tell you that we didn't actually recapitulate that in year 2. So we'll come back to that in a couple of years, probably, or next year. Within that single haplotype, we find some variation within both RNA-seq and GBS. So that's concerning, sort of. And so far, this is, again, our main take-home. So far, it's challenging to link specific genome function to invasion, other than making associations with variation, right? That's still where we are. I think that's still state-of-the-art for this system. Ongoing studies. Oh, and by the way, this was all summarized by Bethany. So it'll be in a book chapter that's coming out soon. It's a really massive effort that she undertook to collect all this stuff. So I'm excited about having it all in one chapter. Right, so we do have ongoing work to report the epigenetic variation. We have a genome for the diploid multiflora. That's a whole different can of worms that I can't talk about today, unfortunately. We have this really fantastic herbarium genomics work going on, and Kyle Kiefer is working on variation in the herbarium specimens in terms of stomatal size as they vary across space and time. And we hope that we'll be able to get to the microbial associations in Bethany's work, but so far it's not looking promising on the funding landscape, so we'll see how that goes. Okay, so just a word about limitations, right? So I've been mentioning that we have limitations. Evidence for that is twofold at this point, my main sources of evidence. So this paper was published a couple years ago. Mark Schmidt is the bioinformatics guy in our team. He's really fantastic. They published a summary of the genome size of plants that had been, they were using in an experiment, and showed that the percentage of the genome represented with their reduced represent bisulfite sequencing ranged from 3 to 11%. And unfortunately for us, with Spartina, we're talking about less than 1%. So in the MOPCA project, we actually quadrupled that by using a lot more resources, and we still have much less of the genome than we'd like. Right, so we're still at like, well, I don't think we're at 20. We're at somewhere around 15%. So you can optimize epiGBS by changing your enzyme choice, changing your choice of organisms so they have a smaller genome size, which is not convenient for those of us who care about certain ecosystems. You can also throw a ton more money at the problem, but we're still really limited in terms of sequencing power. Or you can think about other markers. So I'm often trying to convince people to move away from epigenetics and into gene expression if they have these challenges, because you can get a little more bang for your buck. This is the other fascinating thing about reduced representation and trying to do epigenetics. So what we care about in epigenetics is if the methylation is happening somewhere in a space in the genome, like the promoter or transposable elements upstream of your gene, right? This is what we care about. If there's differential methylation here, you're probably going to get differential gene expression, and that could affect function. What we actually know with reduced representation data is fragments across the genome. We don't know where those fragments sit in the genome most of the time because we don't have a reference genome. Okay, so we talked about some of these limitations in this new phytologist paper here, if anybody would like to know more about the problems. Right, so I argue in almost every talk that I give that most of our knowledge of genome function is in model species. We really need to sort of invest in understanding more complex genomes in complex environments, understanding sources of variation, how much of it's inherited for methylation, the role of methylation in gene function. These are still really unknown and difficult to attack. And the bottom line being, we need to develop genome level approaches in more species of ecological interest. So we've written about these problems and big conceptual stuff, and I just want to highlight that here real briefly. We have this Ecology Letters paper, this Paradimi paper I wrote with Massimo during COVID was amazingly hard, but had a summary of the state of the art at the time. Jeannie also coordinated this review of the role of epigenetics in invasions. And then working with David Lewis and Joel Brown on a couple of different projects, we talked about, and also on the second project with invasive species, working with Drew Kramer and Fargum, we talked about genomic processes and comparisons between ecology and cancer studies and what we can learn from them. I didn't have time today to talk about some of the other studies in my lab, but I definitely want to highlight some of the work that other students have done. So Bethany is still here. She's becoming a jack of all trades. She's actually now working on seagrasses with Susan Bell, funding from Moat Marine Lab and Department of Environmental Protection. Nancy Sheridan is currently at FWC. She was at FWC during her dissertation. She worked on Condylactis. And both of these are assessing sort of restoration and conservation questions in local marine habitats. Teresa was working on mechanisms of heavy metal accumulation and tolerance in mosses. She had a Marie Curie postdoctoral fellowship and was with us for several years. Arish was a PhD student here, although she was already a doctor in cancer at the Moffitt Cancer Center. And she did this fantastic study where she actually evolved resistance to tumor growth in mice. And we worked on looking at the molecular mechanisms that are associated with that. And then, of course, we have this big effort in the department, which Lauren was leading, particularly between our lab and John Parkinson's lab, but also involved several of our other faculty like Diego and Camilo and Mark Margaret, Ryan McMines, who's no longer with us. But this was really fun, and it brought together a lot of our ideas coming from different perspectives. I also want to thank a lot of the undergraduates. This is not all of them, but these were the photos that I had on hand. We've had really fantastic undergraduates working with us, as I'm sure you know. Finally, I just want to highlight that I've been able to establish this really great global network of people, mostly through the MOPGA. So this is our team that finally got to meet each other in person in Germany last summer because COVID had prevented things. And as you all know, like the project started in 2019 and basically spanned COVID. And getting to China was impossible. And even after COVID, for other reasons, traveling between China and here, between China and Europe has been challenging. I'm also working with a big group of the MOPGA scholars to try to come up with this multidisciplinary statement about the need for funding and collaboration and work across disciplines to solve these big global change problems. So this is in review right now, and we'll see if we can manage to convince somebody to let us say some things about our government that we don't appreciate. We've spent some effort working with local journalists to get our work out to the public. So these are available online. The University of Tübingen has their own sort of magazine, which is there. So it's written in both German and English. This is Slate magazine that came to talk to me about knotweed, or actually called me, and we did it over Zoom. We published a paper for kids in Frontiers for Young Minds, which was really fun. These are all MOPCA laureates down here working in various projects of response to climate change. And then, of course, we have Darwin Day. So we published a blog during COVID because we couldn't host any events during Darwin Day. My poor young child, Dorothy, did a drawing for it. She really wanted to. I know she did. And then most of you know about Darwin Day because I hound you about it in February, but it has been amazing to be able to work with the county, and consistently every year we've had a pretty fantastic Darwin Day speaker. We've also had high school workshops that have been organized by IB graduate students working across campus with other departments and other students. And the posters were largely designed by IB graduate students too. So that's been a fun way to interact on outreach stuff. I do want to say particular thanks to Professor Alex Levine in philosophy and Mr. Larry Plank, who used to be the executive director of STEM in Hillsborough County, but has now just moved to Kennedy Space Center to be in charge of their education outreach programs. So if anybody is interested in organizing stuff to go there, he's definitely a fantastic resource. These two guys have been working with me since our first Darwin Day in 2012, and it's been really cool, really fun. With that, I think I've mostly mentioned the various types of funding sources we've had. I need to thank my family, of course, host Oliver Bosdorf in Germany, lots of students and postdocs. It's been really fun to work together, lots of colleagues around the world, and to the department, because I just cannot express my appreciation for what I've been able to do for me and my family. Hello. Oh my God, really? Why does it stop sharing? Did it turn off? Oh, why does it say stop? Oh, that's to take it off. So I had one last little thank you here for the department, but I don't know if it's going to show up. Oh, there it is. So I just wanted to say thank you to the whole department. So the staff, the faculty, I feel like I've been able to do all of this my own way, and it's a really rare opportunity to have that support. So thank you to everyone. And that's it. Steve. You know, it could be. I think any system where you sequence two of the same individual, you're going to get some polymorphism. So there's been some work recently highlighting that. But the ploidy is a different problem, because when we have fragments, we think we're looking at the same part of the genome, but it could be that the polymorphism belongs to a different part of the genome. Right, so the whole problem with sequence data is that you're aligning fragments and you think you're aligning like with like. And then you have to decide, is one polymorphism enough to kick it out to a different part of the genome? But there's also sequencing error, there's PCR error. So there are all these little tiny sources of error that can also be amplified by the time you get your hundreds of thousands of sequence fragments back from the sequencer. So it's really turned into a big statistical game to try to find the good data within the massive amount of data that you get. Definitely improving, with the technology, but it's still like, can I trust this? And for us, I look back on my starry-eyed, naive, oh my God, this is like a giant experiment of taking replicates of the exact same individual and throwing it around the world to see the full range of variation. That was 100 years ago, though. And so I just, didn't think about, oh yeah, mutation happens. I mean, even within your body. So working with the cancer people has really made me also think about, even within an individual, you have mutations. We always say all the DNA in every cell of your body is the same, but it's not, right? So once you start thinking down that path and extrapolating to 100 years of time and ecology and lots of individuals across the landscape, any of those small differences can really add up. Yeah. Yeah, Camilo.
I'm wondering, and I don't know if I don't know how good it is, but to think about the way you put us this planet is everything, and that's why it's significant.
Mm-hmm.
But that's not the rule when you go to look into it. And most plants have functional field, and they will stick.
Right.
Pioneer will do what pioneers do. Shade tolerant will do what shade tolerant do. So on and so on. And probably that's why we didn't have that many stories like all around the planet. That adding massive complications to it because that plant has the battery, the genetic battery to any challenge. And not eager to ask some of those, like the majority of the plants that not go out of.
Yeah, so you're arguing that if I wanted to ask a specific question about how do plants respond, it would be better to work with a plant that's easier to work with. Yeah, I mean, believe me, I get that argument every time, especially when I talk to Arabidopsis people. They're like, "What are you talking about? Oh, why?" But what I argue in response typically is that this is the plant that's having the impact. This is the plant that we want to understand, and this is the plant that's the exception to the rule, which could be really insightful for how it can be done, right? If we could decipher it. So that's why we're trying to attack it from lots of different angles. So understanding that the phenotype is changing, understanding that the genome is gigantic. And I didn't have a chance to talk about it today, but we're also trying to dissect the origin of that genome in terms of which diploid eventually turned into this giant octoploid. And that's another big question that we don't know. But it's like a game of chance. It's like, what are the chances that you're going to get this genotype, this individual genotype probably in the beginning that is just so able to deal with all this stuff. So this project, this paper where we found the ramette production is higher in Japan. So then we're thinking about that trait was already there. These plants in Japan are not as tall, but they make more ramettes. So that trait was already there before the guy chose that plant, right? So then we're trying to dissect what was it about that plant that allowed it to be invasive. So yeah, I mean, I get it that there are different ways to ask specific questions about which traits are, how different traits are going to evolve, how different plants are going to respond to environment. But I think the question here is more, why is this thing able to do what it's doing? And then we can maybe come back and compare, this plant makes more ramettes than these other populations. Is the ramette production important more generally speaking, right? When is ramette production important? Why are the Japanese plants more ramette producing? Like, those kinds of questions. So this is just part of it that's interesting to me. But it's kind of a plague that it's got such a high ploidy and a giant genome. we're just really limited. So, but at the same time, every part, every little tiny piece that we find is still findings, baby
Yeah,
steps.
no, it's fascinating. So,
Not what you're interested in. Like, I wouldn't do it that way. It is. It is.
that's
Yeah. But it's also true in Spartina. I mean, Spartina has a ton of polymorphism, but it also has a giant genome. And you can't deny that Spartina is a massively important plant. It makes up a huge portion of the intertidal biomass up and down the East Coast. And that's a really hugely important ecosystem. Like even just the natural ecosystem, right? So Japanese knotweed's a little bit frustrating because it's an invasive. So it's, trying to make the argument about how important it is a little harder. So it often comes to how are you going to apply that to killing it, getting rid of it, which I have no idea because I don't care. But the Slate magazine is funny because the author wrote in there, and then I started talking to Dr. Richards, and she didn't care about getting rid of it. She just thought it was cool. So, yeah, that's my fate, though.
very nice talk.
Thank you,
I
John.
ask you about your haplotype network.
Oh boy. Yeah.
Well, no, not too in depth.
Okay.
It's all based on chloroplast.
Yes.
So do you think maybe that's masking? Like maybe you need a better marker.
Yes. It's a really good point.
Not as extreme as what you're saying in, say, your whatever the polyploidy is contribution.
Yes.
But maybe there's a nuclear marker that would be more representative of reality.
Yeah, it's totally a fair point, and it's really astute. Those of you who aren't catching the question, those are based on chloroplast markers, which means that it's a maternally inherited marker. So you're basically only tracing the mom through time, right? And the reason that we chose that is because it's much easier to do technically in the lab, and also there's other published chloroplast genomes, so that we could lean in a bit on what other people had already done. But for sure, the diversity that we're seeing is probably more represented, and that's why we're doing GBS. But then, yeah, so it's this circular problem. But the nice thing about the chloroplast markers, at least recognizing the limitation that it's maternal, is that we were able to confirm the overlap with the Kyushu populations. So this was a thing that people had talked about. We know von Siebold brought those things from that part of Japan. It's documented in letters and things, but to have the molecular evidence from all of those populations that they are in fact most likely descended from that area of Japan was nice to see. And this was one of those projects, like I didn't have any idea that this is something we would do. And then we hear from our colleagues in China that they wanted to do it, and they just ran with it, and it worked out really nicely to confirm what we always say we know, and now we do. But yeah, the GBS will hopefully be more informative. I mean, like I said, we spent years just trying to figure out, can we trust this data? Why is it telling us there are so many polymorphisms? Like, why? So yeah, we're still working on that. We haven't been able to do the tree with the GBS data yet because of that. I mean, I think we have a tree, but I'm always looking at it like, is that going to change if you move that red line, And we can't just go and do triple and quadruple sequencing of every sample. And we definitely can't do whole genome sequencing of every sample. So yeah, it's a fair point though. Thanks. That was 66 slides in like 50 minutes.
What do you think about the term multidisciplinary versus transdisciplinary?
Oh my God, wow. Yeah, so Dr. Kramer just asked how I feel about the terms multidisciplinary versus transdisciplinary versus interdisciplinary versus blah, blah, blah disciplinary. this is a real problem. We sent this article to PNAS, and one of the comments was, this is not multidisciplinary. Like, people are invested in this terminology, right? And it's not, because we have in MOPCA 53 projects across a huge number of disciplines. But there wasn't really, we had exchanges, but it was like beginning, middle, and end kind of conferences where we all sat and talked about what we're doing. It was each person talking about their work and all of us listening. So it wasn't like we broke into groups and never talked amongst each other. But none of the projects were originally created to include multiple disciplines in the project. They were chosen by different disciplinary experts, and the program as a whole represented all those disciplines. And by the time we got around to actually talking, this was during COVID, of course, so we had limited interactions anyway. One of the take-home messages is this should be from the beginning a multidisciplinary effort of coming up with projects that actually include the social sciences as well as the different sciences to actually make progress the way that we think we need to make progress in addressing global change questions, right? Because they're inherently going to involve a lot of social issues. And how do we study that? We just don't. I mean, I certainly don't. So I think it is a problem. I think what's worse is that the language is really hard to dissect. People write about using those terms, they write about different versions of what they mean. So we spent a lot of time trying to figure out, what are we? And I don't even remember the answer because it was one of those kinds of discussions, but we had to change it. But then, well, I can't tell you. Not because it's a big secret, but just because I don't remember. Because in my mind, I don't think about that kind of stuff. And so I just listened to everybody's opinion. We read the reviewers' opinion, and we tried to find a better term. And we do have sociologists working with us, social scientists who are like in energy policy and social issues stuff, which is super important, and I really care about it, but it's language specific that's really hard to bring together. So those guys were like, oh yeah, we need to use blah, blah, blah. And I'm like, done, cut and paste, stick it in there, and that's fine with me. I mean, we weren't trying to say that MOPCA had done that. We were trying to say that that's where we need to go next. But writing this paper is hard. And the last comments we got from the editor at PNAS was like, this is not the right place to be attacking the current president of the United States, which is exactly what we were doing, right? Because we had a draft before the current president got elected. Then he comes in, pulls out of the Paris Agreement again, and starts attacking the scientific infrastructure of the country. So then we were like, and now that we have another round of this, and we were really specific in the paper, and PNAS was like, no. So it's a really interesting new experience for me, not to mention that you might have seen there are 80 authors on that paper. And I'm the one that's like, we're gonna do this. Yeah. Which I guess you saw also in the Nineveh paper. It was the same kind of like, we're doing this. I know it's hard, but we're gonna do it. Yeah. So thanks for asking. It's a good question. Yeah.
So I'd like interested like enemy hypothesis.
Okay.
I was just curious because you showed that slide that there's more like pathogenic and
In China. Mm.
So when you did the common garden experiment, you see differences.
Oh my God, this is a big can of worms. I would love to have a longer chat with you about it. The bottom line is there are differences. So we published a paper in Ecography that I did not include here because it's so messy. But there are differences between the attack on the Chinese versus the introduced, and also which guilds, I guess they're called, different types of insects that attack. So those differences are real. But what's really strange and hard to understand is you would think that the plants that escaped to the introduced range and lost their ability to fight off the herbivores, when you take them back to the introduced range, they should. That's not what we found. So we can't make that happy story about the enemy release hypothesis now that we have common garden data. We can only make that happy story if we look at the field data. It gets really complicated when you actually have the evidence that, oh, actually, no, they're not less defended. Yeah.
Afterwards, I'll get the paper.
Yeah, for sure. Because I really don't know what to make of it. Because I look at that, and I'm like, that didn't work the way we thought, the way we thought it did. But thanks for asking. Yeah, I think we're done, guys. Thank you so much for coming. That's awesome. You let me just like blast away for an hour and 15 minutes. Sorry.
So this like a tiny frustration among all the administrators.